video, this live, I guess. Which receptor ligand interaction enhances MHC1 expression on nucleated cells? I literally just guessed on this one. It's interferon alpha or beta to type 1 interferon receptor. Why? It's because type 1 interferons, alpha and beta, upregulate MHC1. So think type 1 is alpha beta, type 2 is gamma, and then type 3, I don't know, I'll probably get to that though. Which of the following are granulocytes? Neutrophils, eosinophils, and basophils. Which immune cell is the primary component of pus? Neutrophils. They dominate purulent material in affective tissue. Another word for purulent material is pus. Which cells release web-like DNA traps to immobilize pathogens? Neutrophils do. They literally just like explode. They go to an antigen and they just throw all their DNA at it, which is good. It damages the antigen. Helps get rid of it. But it also gets rid of the neutrophil. Which cells produce nitric oxide via INOS? M1 macrophages. Remember that M1 macrophages are the hostile kind, or the hostile setting, if you will, whereas M2 is the healing, the repair setting. Oops. Okay, and then which cell provides the best antigen presentation? Dendritic cells, because they are professional APCs that activate naive T cells. Which cell type is resident in tissues and similar to basophils in function? Mast cells. They're stationed in tissue and respond with histamine release. What structure allows lymphocyte entry into lymph nodes from the blood? High endothelial venules. I actually got this one wrong. I put afferent lymphatic vessels. It's because HEVs, or high endothelial venules, specialize in recruiting lymphocytes from circulation into nodes. What's the difference between natural killer cells and CD8 T cells? Well, natural killer cells don't need MHC presentation. Which surface marker identifies natural killer cells? Hi, how's it going? CD56 is the surface marker on NK cells. Afferent lymphatics deliver to what nodes? They deliver to lymphocytes, APCs, and antigen. Sorry. Afferent lymphatics deliver what to nodes? They deliver lymphocytes, APCs, and antigens to the nodes. Okay. Which of the following correctly matches the type of interferon with its main source and function? I guessed on this one too, I think. Type 2 interferons are produced by T cells and natural killer cells, and they activate macrophages and they boost antigen presentation. Type 1 interferons help protect surrounding cells from viral infection primarily by inducing antiviral proteins and increasing MHC1 expression. This is immunology, veterinary immunology to be exact. I'm just studying for vet school right now. And me teaching other people helps me study. But also, it might help other people learn. Maybe they'd find something that's interesting. Thank you. All right. Coding viruses. So Yeah, they induce antiviral proteins and increase MHC1 expression. Type 1 interferons induce an antiviral state, increasing MHC1 and inhibiting viral replication in neighboring cells. So, tumor necrosis factor alpha induces fever and acute phase response. You're from Mexico? That's really cool. That's awesome. Okay, and then the primary function of type 1 interferons, which are alpha and beta interferons, is they induce antiviral states in neighboring cells. Basically, they warn nearby cells. Yeah. The cytokine that is critical for lymphoid organ development is tumor necrosis factor beta, which is also referred to as lymphotoxin alpha. Now, which statement about interferons is true? They alter cell states to resist viral infection. Upregulation of MHC class 1 molecules is most associated with which interferon? Type 1, which is alpha and beta. Which of the following best explains why interferons are critical in viral defense? They modify the intracellular environment to resist viral replication. A fundamental mechanism by which interferons exert antiviral effects is by altering the cell state to resist infection and enhancing immunity. Tumor necrosis factor alpha is primarily produced by macrophages, T cells, and natural killer cells. What is the main site of B cell follicles and the germinal center, main site of B cell follicles and germinal center formation in a lymph node? It is the cortex. Which cells dominate the paracortex and what happens there? T cells do, and antigen presentation happens there. Why do lymph nodes have many afferent vessels but only one efferent vessel? What it does is it slows flow and it maximizes immune screening. So, natural killer cells are part of which immunity arm? And do they provide memory? They're part of innate immunity, and no, they do not provide memory. Which cytokines activate natural killer cells during viral infections? Interferon alpha, interferon beta, and interleukin, I think that's how you say that, 12. One mechanism by which NK cells kill targets is antibody-dependent cellular cytotoxicity, or ADCC. Natural killer cells are they are a type of immune cell. What they do is they go to the molecules in the blood that are found in the blood, antigens, blood, any cell. They go and they look for certain receptors or molecules present on these other cells. So if these other cells have an MHC1, which is a major histocompatibility complex class 1, then the NK cell will be like, oh, okay, that's a friendly cell, I'm not going to kill it. But if the cell doesn't, then they'll be like, okay, this cell's foreign. But there's other ways that, well, there's I don't know, there's a little bit that I can't like regurgitate right now. I can't speak about, but they kill cells by perforins, granzymes, and then they literally cause the cell to blow up, which is called apoptosis. So yeah, they're pretty cool immune cells, very crucial. Lymph node compartments are the cortex, the paracortex, and the medulla. You're welcome. The lymph node versus the spleen. The lymph obviously has to deal with the lymphatics, the lymph, right? The spleen has to do with blood. Afferent lymphatics deliver what to nodes? They deliver lymphocytes, APCs, and antigens. APCs are also called antigen presenting cells. Yes. The sequence for a natural killer cell when it's killing a foreign cell is it attaches, it uses perforin, it then uses granzyme, and then it causes apoptosis. Where does positive selection of T cells occur? It occurs in the thymic cortex. What is the fate of most developing T cells in the thymus? To die by apoptosis, actually, in a process called negative selection. Yes, okay. What is the purpose of negative selection in the thymus? To eliminate T cells that strongly bind self-antigen, because we don't want those kinds of cells. Those are going to hurt our body. Which thymic structure is associated with negative selection? Hassel's corpuscles. Those structures support self-tolerance induction in the medulla. Where do I recommend studying for medical school? Well, it depends on where you live. I would look for good medical schools in your area that are local in the same state that you're in, because it's cheaper that way. And usually the education's the same. But med school is a little different than vet school. I'm not in med school, I'm in vet school. Similar in a lot of ways, very, very similar, but not the same. Is it hard to get into? I've heard, yeah, it is. It can be challenging. Vet school was very challenging to get into. I was fortunate. I was able to do something called the early admission program, which means I basically had a straight shot into vet school from undergraduate. But the catch was that I had to keep above a 3.5 GPA in every class, and well, no, it was actually per semester and overall in undergraduate. So I had to have like all A's and maybe one B a semester. Maybe two. It was pretty crazy. In calves and lambs, the ileal Peyer's patch functions as the primary lymphoid organ for B cell development. Only in young ruminants, and I don't need to know that for the exam. So, where do T cells undergo positive and negative selection? The thymus. Because T cell, whenever you're thinking T cell, think thymus. Okay, then we have, in most mammals, where do B cells mature? They mature in the bone marrow. Think B cell, bone marrow, right? So for vet school, well, really in vet school, so GPA is very, let me see here. So an A, so if you have an A in every single class, that's a 4.0 GPA, which is the best you can do, really. I mean, whether it's a 100% in every class or a 90%, as long as it's an A, that's pretty much how it works in America. But a B is a 3, a C is a 2, a D is a 1, and then an F is a 0. A failing grade, you don't get any points. In vet school, all we need to graduate is above a C in every class. So thankfully, I don't need to be stressing about, oh, I need an A, or oh, I need I can only have one B this semester or else I'm kicked out of the program. No, it's not like that. Not like that anymore. It was like that, and that's only because I was going from undergrad into a very prestigious program, and they were kind of trying to test my mettle. They were trying to test and see, does he really is he going to make it if we put him in vet school, or is this just a waste of time if we do? And thankfully I got in. But now I kind of just got to keep above C's, thankfully. Very thankful for that. And I don't have anything after this. I don't plan to. I'm not gonna try and get a PhD. I'm just trying to get my DVM, which is Doctor of Veterinary Medicine. But I'm so glad that undergrad is over, because yeah, that was stressful, keeping really good grades, because some of those classes were really challenging. They were fun, and some were easy, but not all. A lot of them were really challenging. Is it tiring studying for this? Yes. I've been studying this for at least an hour now. I just got done with my other live stream, which was for a separate day. What I'm doing is I'm like going day by day, studying everything that I learned those days. And yeah, it is tiring. It takes a lot of time and effort. But not as much as I thought. I thought I would have no social life. I thought I would have no time for entertainment like video games or anything, which I definitely have decreased time. But in order to, have fun every now and then, or, I mean, it's either that or I just study 24/7, don't sleep, don't eat. That's no way to live, right? You've got to eat, you've got to sleep. It's hard, but it's not impossible. If it were impossible, veterinarians would not exist, right? But yes, it's very tiring. What is the purpose of positive selection in T cell maturation? Well, the purpose of positive selection is so that T cells can ensure self-MHC recognition so that they don't kill host cells. Okay. And also so that they can attack antigens that have their materials presented to them by the MHC. After activation, what can B cells differentiate into? Plasma and memory cells. Which primary lymphoid organ matures T lymphocytes? The thymus. Again, T cells, think of thymus cells. B cells, think of bone marrow. Now, I can't remember if the B cells are made in the bone marrow, but that's where B cells mature. Which of the following best describes MALT, which stands for mucosal associated lymphoid tissues? Well, mucosal associated lymphoid tissues are unencapsulated lymphoid aggregates at mucosal sites. Tonsils, Peyer's patches in your intestines, something in the lungs too, I forgot the name of. Which mucosal lymphoid tissue is paired incorrectly with its location? There's a list here. The thymus is not found in the oral cavity. It's found near the, or it's found in the thoracic, it's found in the thorax, and it regresses as someone gets older. But these other options here, let's go through them here. GALT is found in the gastrointestinal tract, gastrointestinal associated lymphoid tissue. BALT is found in the respiratory tract. I don't know what the B stands for. NALT is found in the nasal cavity, probably nasal associated lymphoid tissue. Yeah. Which antibody type dominates MALT immune responses? The correct answer is IgA. I got that one wrong. I put IgM. I don't really know what all they do, to be honest. I better know, hopefully, though. What type of specialized epithelial cell facilitates antigen transport to lymphoid tissue in GALT? The M cell does. M cells in Peyer's patches transport antigens to immune cells. The rationale for that previous question, by the way, secretory IgA is the key antibody at mucosal surfaces. Good to know. Type 1 interferons, like alpha and beta, are produced by most nucleated cells during viral infection. Type 3 interferons, which are type gamma, interferon gamma, primarily function at which site? They primarily function in the epithelial surfaces, such as the gut and the respiratory tract. Mucosal surfaces. Which cell types mainly produce type 2 interferon? Sorry, type 3 interferon is interferon lambda, not gamma. Interferon lambda. Type 3 interferon lambda. And they work in epithelial surfaces such as the gut and respiratory tract. Type 2, which is interferon gamma, are mainly produced by natural killer cells and T cells, NK cells and T cells, especially in response to intracellular pathogens. The main role of type 3 interferon is providing local mucosal antiviral protection. Interferon lambda. Which immune functions are enhanced by interferons? Activation of NK cells and increased MHC expression. And then finally, the last question. Excessive tumor necrosis factor alpha signaling can result in septic shock due to uncontrolled pro-inflammatory responses and endothelial activation. What it does is it causes vascular permeability and cytokine cascades, which leads to shock. Finally done with that. Now, I'm going to move on to my immunology notes. So that was my immunology practice exam for my exam tomorrow. Now there's a portion in my notes that I am going to review here. Let's see if I can find it. Okay. September 3rd. I'm very behind. Neutrophils are, I don't know if they're granulocytes. That's what we need to know. Made in the bone marrow. They've got a short lifespan. They're in the circulation until they're needed, and they die pretty quickly. They never go back into the blood circulation once they leave the blood vessels. They basically just phagocytose microbes. Phagocytize? I don't know. Anyway, They're the first line of defense in the fight against bacterial infection. It is phagocytose. They're first summoned by cytokines produced by sentinel cells before phagocytosing bacteria. They're toxic for tissues, though. This is why if you have an infection and you have a lot of pus in there, it hurts. It's all inflamed and red around the pus. That's because the neutrophils are damaging your tissue, literally. But it also gets rid of the bad cells in the infection. Similar compounds to bleach. They don't live very long. They're kind of like Spider-Man, webs. They shoot traps. They've got a limited energy reserve, limited number of phagocytic events. They go back to the bone marrow if they're not needed. Then they undergo apoptosis and are removed by stromal macrophages. Okay, then macrophages. Many different kinds. Osteoclasts in the bone marrow, microglial cells in the brain. You've got type 1 and type 2. Type 1 is the hostile version, pro-inflammatory. Type 2 is the healing version, anti-inflammatory. They've got many different kinds of receptors. Receptors like FC receptors, complement receptors, CD40, MHC1, MHC2, pattern recognition receptors. And their phagocytosis is similar to neutrophils, but macrophages are also attracted by elastase and collagenase, enzymes that are released by dying neutrophils. Macrophages are activated by interferon gamma, which is produced by natural killer cells and T cells. Yeah, I don't know why it's TH. Thymus, probably. Thymus cells. Again, interferon gamma, think. That's type 2 interferons. It's kind of like they're on a thermostat, those macrophages. And if they're activated, they become hyperactivated macrophages. They increase in size, movement, activity. They can engulf whole cells, that sort of thing. They destroy pathogens. They make nitric oxide, which is toxic to the body, but it's in such small doses, and they use it in a certain way that's only toxic to the pathogen, basically, I think. That nitric oxide can efficiently kill bacteria, fungi, protozoa, some helminths, which are like worms, and even tumor cells. So cancerous cells. Their macrophages are necessary for wound healing, and they secrete proteases which break down connective tissue. They regulate collagenase and fibroblast activity. And once they remove the damaged tissue, they secrete growth factors for collagen synthesis by fibroblasts and for growth of new blood vessels. Think of scar tissue. It fades over time because your body's literally remodeling. Okay, again, M1 and M2 macrophages is kind of like attack mode and healing mode, respectively. Tumors and pathogens can sometimes control the environment to make it seem safe so that the macrophage is in M2 mode. That's how cancer happens, and the cancer cells are not killed. Eosinophils, mainly functioning for parasites and allergies. Yeah, okay. Natural killer cells, CD56. They destroy virus-infected cells, cancer cells, and cells infected with intracellular protozoa and bacteria. So natural killer cells basically do kill host cells, but only host cells that are affected. NK cells are also lymphoid. They're not myeloid, they're lymphoid. They're activated by interferon alpha and beta. Okay. They basically just examine abnormal cells. Yeah, I'm getting tired or something. They look to see if the MHC is missing, basically. Activating receptors, inhibitory receptors. Man, I'm so done studying. I'm getting pooped. Inhibitory receptors override the activating receptors. And NK cells kill via ADCC, antibody-dependent cell cytotoxicity. They also kill via the perforin granzyme pathway, which perforins perforate the cell. They form pores in the cell membrane, and then granzyme enters the cell and it binds to cell proteins, which induces programmed cell death or apoptosis. Innate immunity. Okay, that's it. That is all for immunology, and that's all for the 3rd of September. Thank you all for watching, and I'll see you in the next one, hopefully.